Thank you very much for the invitation. So yeah, I'm Irene Papatheodorou. I lead the gene expression team at EMBL-EBI. So first, I wanted to talk a little bit about what is EMBL and EBI. So basically, we're an outstation of the European Molecular Biology Laboratory in Hingston, which is just outside Cambridge. And basically, we are a world-leading resource of public biomolecular data. Our vision is to benefit humankind by advancing scientific discovery and impact through bioinformatics. We do lots of things at EBI. So we deliver data resources, and I'm going to talk about one of them. We perform excellent research. We train next generation of scientists in all aspects of bioinformatics, engage with industry. So we work a lot with pharma companies and others as well. And we also coordinate bioinformatics in Europe through projects such as Elixir. So as I said, I'm going to focus on one service, but we have lots of different services at the EBI to support all aspects of life science research, starting from chemicals and molecules and drug discovery. You can see the types of resources we have. Genes, genomes, and RNA, and I'm going to talk about expression atlas, that's in the middle there, proteins, imaging, and cellular structure, genetic variation and disease data, and literature and knowledge management. So our databases, because the communities we serve are very collaborative, and we are collaborative as well, we have lots of different interactions between the different resources. This image shows the internal interactions between resources at the EBI based on the exchange of data. So when a resource has a release, feeds data into other resources and enriches them. And basically the width of each internal arc is weighted according to the number of different data types that are being exchanged between the different resources. So expression atlas that I'm going to talk about is over there, and it connects to lots of different other resources. What is the expression atlas? And then I will talk a little bit about the single cell expression atlas. That is the latest addition to the resource. So basically the mission of my team is to provide to the scientific community freely available information on the abundance and localization of protein and RNA across different species and biological conditions, so that any researcher can query and ask questions such as where is my favorite gene expressed, where in the body, and how its expression changes in a disease, for example. So I thought I should do this since this is an informatics department. So basically, what do we mean by gene expression and protein? And starting from the DNA that exists in each cell, when there is a certain environmental factor or based on the housekeeping functions of the cells, particular genes are being turned on, as we say. So RNA is produced for these particular genes, and then through a process called transcription. And then from this RNA, protein is being produced through the process of translation that actually does the job. So what we measure really for the experiments that we take in is the RNA. So we know which bits of DNA are being active, they're being expressed. We don't measure the protein, so it's kind of an approximation of that. But when I talk about gene expression, I mean measurements of RNA levels. So just to give you an idea of the types of experiments that we include in Expression Atlas, this is what we call a typical baseline expression. So say you have a tissue like heart, you take a sample, and then you isolate the RNA from there, and then you find the bits of RNA that correspond to active genes. You sequence that, and we get the big files with the RNA sequences that show the transcriptome, which are the genes active in that tissue. And what we do is we perform analysis to identify which are these genes, how much they are expressed, at what levels, We do various things of normalization, batch correction, and so on. And then we get a result of the particular genes that are expressed in the heart. So the other type of experiment we deal with is differential, where instead of just having the genes that are expressed in the heart or some other tissue, here we ask a question like, how does the gene expression change? For example, with infection of COVID-19, right, in colon or lung. And we have a sample of a healthy person and a sample of a diseased person. And then we do all the process that I showed before, but then we do a differential expression between the two samples. And then we find which genes are upregulated in the disease condition and which in a particular tissue, and they're in colon. And here, which genes are upregulated, some are downregulated in the lung. So these are the two main types of experiments that we deal with. And the aim of my team is to take all these kind of data sets from archives that are being produced by the research communities and curate them, perform systematic analysis, and then extrapolate the gene and protein expression across the data sets from the public domain and calculate what is the abundance and localization of the genes. And there are lots of challenges doing this. So first of all, in the public domain, there are data sets from different conditions, developmental stages, genders, tissues, and so on. They're not always comparable. And also we need to bring together data sets that have been produced by different labs, using different assays or technologies and so on, that introduce lots of batches that need to be corrected. So the first challenge, we do organize the studies using ontologies and the metadata that are associated with them so that we can search and combine them and then find out which ones actually could be analyzed together or not. And then in order to tackle the second part, that we have data from different labs and so on, we use standardized analysis pipelines so that we perform the same kind of analysis for the same kind of data and at least reduce in this way any errors. And also we perform batch correction at the end so that we can facilitate meta-analysis across studies. So this is the data journey in my team. First of all, we collect, we handle the submissions of all the gene expression and functional genomics data at the EBI, and we curate them. And we also, within that process, we standardize the curation and ontology annotation processes as well. And also we collect data from other resources such as GEO, the Gene Expression Omnibus, and ENA, and so on. Then the second part is we build the analysis pipelines. So we have transcriptomic data analysis pipelines for bulk, single-cell RNA-seq, also microarrays. And we have standardized, especially for single-cell RNA-seq, which is the most recent, all the steps of the analysis from mapping to the genome, quantification of the gene expression, normalization, clustering, and cell type annotation. And we have productionized this pipeline so they work pretty much 24/7. When a curator pushes an experiment, it gets picked up, gets analyzed, and then loaded to the staging version of Expression Atlas. And then we also develop the user interfaces and the necessary databases to make this gene and protein expression easily accessible and reusable. I keep saying protein expression as well. So that's because we collaborate with the PRIDE archive, which is the protein archive at the EBI. So all the proteomics data that measure protein expression levels get archived there. And that team has developed their own standardized analysis pipeline. So we feed this data into the Expression Atlas as well. And the idea of having all this data there in an easy to use way is that researchers, biomedical researchers who cannot really do the analysis, they can easily find this information in a way that is interpretable, and it can drive new research hypotheses. So this is the front page of Expression Atlas. That's the link down there. At the moment, we have collected over 4,000 bulk RNA-seq datasets. Bulk RNA-seq datasets are the ones where you take tissue and you get the gene expression for that tissue, right? And that's about 300 baseline expression datasets and over 4,000 differential expression datasets coming from 65 different species. And you can read more about it on our update paper for NAR. So what does it look like when someone does a query? So here someone has, that's me, has queried the expression of a gene called reg1b. And what they find is on the front page, on the first query results page, is a heat map of the different tissues, which is on this axis, and on the y-axis is the different experiments where this gene has been found to be expressed. And what one can see is that it's highly and consistently expressed in one particular tissue where the blues are darker, and that is pancreas. And also the proteomic data that are at the bottom also agree with the transcriptomic as well. Then when someone looks at the differential expression results, by looking at the metadata, each line is a particular study where this gene is up or down regulated. When someone looks at the different metadata and the comparisons, they find that it's consistently down regulated in pancreatic cancer. So now this has been the bulk side of Atlas that has been around for over 10 years. I want to talk a little bit about a new type of experiment, which is not so new anymore, but talk about the kind of so-called single cell revolution. So, so in recent years, so since I think 2010 or so on, yeah, a bit later. So we have been, researchers have been able to dissociate cells from tissues and then measure the RNA in each individual cell. So basically, this is a nice analogy to understand. So with traditional bulk RNA-seq, what we have is the tissue, which is a mixture of cells in different proportions and different cell types in different proportions. And what we get is the average of the RNA levels across all these. But with single cell RNA-seq, it's like starting from the fruit, where each cell is, you can think of it, each cell is a type of fruit. And then you have the expression in from each different cell. And you can understand the differences in variation in gene expression across cell types. You can also get a feel of the abundance of the cell types in the tissue that might also influence what you see on the bulk RNA-seq. So just to show you how this technology has advanced in recent years. So in 2009, there was the first cell that was sequenced, that the RNA was sequenced. And then the technology has experienced a dramatic development. So in 2017, with especially the 10x genomics technology that is being used routinely now, you can sequence almost 1 million cells from one particular sample at a time. So you can get a pretty good coverage and discover even rare cell types. So that actually encouraged the community to build the Human Cell Atlas. Basically, this is a huge collaboration an international collaboration to create a comprehensive reference maps of all the human cells in the body, in order to be able to understand human health, diagnosing, monitoring, and treating disease. And I think so far there have been over 17 million cells sequenced through that project. And it's a huge coordination effort to bring this data together and synthesize the first draft of the human cell atlas per tissue. So the idea is that we can also understand what each cell type does and what are the rare types of cell types, what is the difference between cell stage and cell type, understand cell fate, how they differentiate, and so on, and also build a kind of periodic table of the human cells. So inspired by all these, about this technology and the results that are coming from the human cell atlas, as well as from other species as well. Now, all mouse also has a cell atlas, and fly, and so on. So my team developed a single cell expression atlas, which is like a daughter resource of the expression atlas to deal with these kinds of data. So just to show you, before in the bulk expression atlas, if we query for reg1b, we find that it's expressed in pancreas. It's downregulated generally in pancreatic cancer. When we have data from the human cell atlas from the pancreas, we actually find, so this is a UMAP plot of the different cells in pancreas, colored by their cell type. We actually found that this gene is expressed really highly only on one type of cell, and that's the acinar cells of the pancreas. So the idea of the single cell expression atlas is that I can bring in these data sets and show to the users in which cell types their favorite genes are expressed and how they change, their expression levels change in a disease or in some other condition. So furthermore, what we are developing is we have some illustrators who develop together with consultants and doctors this kind of diagrams. This is, for example, an anatomogram of the pancreas, where you can zoom in and then show on the right-hand side a heat map of the different cell types within that section, and their marker genes, like their top, the top expressed genes that characterize this cell type, just to make it easier for the users to go through the data. And that's kind of zooming in and showing what the acinar cells look like. These are all ontology annotated diagrams so that we can also interoperate with other resources of the human cell atlas as well, that show different kinds of data like images and spatial transcriptomic data as well. So the single cell expression atlas at the moment has data from 21 different species. That's about 355 single cell data sets, and that amounts into over 10.5 million cells that have been analyzed, processed, and are available for searching. And you can directly find it by the link below. I'm almost coming, I think, to the end of the talk, but in order to develop the single cell expression atlas, we had to develop analysis pipelines that are standardized across the different types of data sets. And I'm not sure if, it is of interest. So basically we use community developed tools for the different parts of the analysis. So the first part that is available through the Nextflow pipeline manager basically corresponds to the read filtering, quantification of the data, and aggregation of the different libraries and samples. And we have two different versions of that pipeline. One is to deal with the droplet-based study. So this is to do with the experimental the way you do the single cell assay. There are two main ways of doing it and require different types of analysis. So we include that. And then what you get at the end here is the raw counts table with all the cells. So basically per sample, you have a 2D table of the different cells and the different genes, and you have values of the expression levels of these genes. So that's what you get. And then we have a further downstream analysis, such as filtering the cells, because many of them are empty and so on, normalizing across the different cells and samples, doing some dimensionality reduction so that we can find more easily which cells are related to others. Then we cluster the cells so that we can find out which cells are similar, come from the same cell type, and so on. And once we're confident with the clusters, we try to identify what cell type each cluster is. And that we do by detecting these gene markers that are particularly unique per cell type. So yeah, and we also have these tools in a user-friendly format workflows, and they're also scalable. They can be run on the cloud via Galaxy, which is it's a pipeline manager with a nice front page as well, an interactive one. So and another thing we've done working with the Human Cell Atlas community in particular is really to look closely at these single cell transcriptomic experiments and decide what is the minimum information about these experiments that is needed for them to be reproducible, for the analysis to be reproduced as well. And basically, this is like a workflow of how of the different metadata involved at the different step of the experiment. And this is what we came together for and request basically for all archives to, for all submitters to archives to submit this kind of information so that we can reanalyze the data. Finally, I'm just going to finish with like future goals for my team in particular and the Expression Atlas. That might be a nice point for discussion here as well. So basically, there's an increasing need from different research communities to use our data for AI applications so that they can combine it with other types of data and infer new relationships, whether it is between disease and gene expression or protein expression or human development or mouse development and so on. So we are looking for these kind of collaborations so that it can help us make our data AI ready and learn about the different formats and the tools. And also, we would also like to invest as well in these tools to extract biological meaning from data, such as dealing with problems such as automatic cell annotation or inference of disease trajectories from the single cell data, different disease stages, and so on. We continue to work with the archives and broker these high quality submissions of these kind of data sets. And there we want to benefit from advances in text mining and so on to speed up our curation and make it more effective. And we continue working on scaling our workflows in the cloud and adding new functionality by adding new analysis. And then the other kind of work we're doing is more on really the data analysis and UI. At the moment, users can only see one experiment at a time in depth, but we started meta-analyzing experiments so that we can uncover this kind of biology-centric information rather than study-centric. And that will be coming into production in the next couple of years. We are also working with the Human Cell Atlas to develop this human-centric single cell expression atlas that is different to the other species by adding multimodal data. So, for example, spatial data and images as well that are being produced by the Human Cell Atlas. And we're also trying to find a nice, easy way of representing the Human Cell Atlas data to the biomedical community in particular, because it's quite difficult for them to really go through the UMAP plots and understand what is going on. And then there is a new push from different other partners to work on a biodiversity cell atlas. So really to make cell atlases of all the different plants and creatures on Earth. So that will be another challenge we will be working on. So I would like to thank my two teams. So the gene expression team that develops the resource I showed now, and the small research group that I run in parallel, our collaborators and funders. Thank you very much.
Thank you very much, Mimi. Do you have any questions? Dimitri? Yes.
Okay.
Thank you. I've got a couple of questions. I will ask one and then the others can ask, and then if there is time, sure, the second one. Okay, one question is kind of a low-level question. It is basically your newest work is to move to single cell analysis, to expression to be particular in different tissues. I wonder, okay, this is very interesting. This is wonderful. I'm not a biologist, but I'd like to know if it matters other things like the age of the person or the gender of the person, or perhaps other factors, let's say the race within quotes. I don't know how the sample is picked up for for the tissues to analyze.
So yeah, that's a good question. So all these things matter. It depends on what you're looking for. So on the single cell side, most of the data are really coming from the West, the Western world, basically Europe and US. Although there is a push now to include more diversity of data from other regions as well. So there's now a Human Cell Atlas Asia and Africa and South America as well. And there are also, so we will see more data sets coming from these populations so that we can uncover the differences in cells and cell type expression across the different populations. Whether they come from which gender and so on, this is already recorded. So there's a whole data coordination platform for the Human Cell Atlas, where they record this information that comes from the different labs, things like age and whether they have some health conditions and so on.
Okay, thanks.
Thank you. I think my question is kind of related to it. It is not very clear to me how the data are produced, not actually produced. The data, I guess, are produced for specific experiments that a researcher is interested in doing. So I was wondering, after the normalization and all this process that is done so that we can have them in a homogeneous data set, let's say, are we able actually to study a new question based on this data, or the major benefit is that everyone can access the same question, the data for the same question?
Okay, yeah, the idea is that you can answer a new question by having the data all together. So for example, the first thing you can ask is my gene consistently expressed in studies that we have that study the same tissue? And if it's not, if some studies didn't find it to be highly expressed and others did, then that actually, makes you question the results as well. So that's one thing. Another thing that we have seen people using is downloading the data from related studies and then applying another tool to answer a different question. So we have seen actually papers that cite Expression Atlas that use machine learning to, for example, combine gene expression with chromatin occupancy data to find how genes are regulated. So we do see that the data are being used to answer other questions as well, even if it's not through the web interface.
Can I ask also a question related to that? Concerning the source of the data, you said that there are studies that have been published, I guess. So concerning the quality of the studies, because probably you have, different qualities on the study, this is somewhat curated by you, or is how you deal with it?
Yeah, so there are different steps. So a curator, when they find these studies in the archives, they first check that they have at least 3 replicates. So we don't take studies that don't have enough samples because we consider them of low quality. And then they check that they have all the necessary metadata. And then we have a quality control pipeline that looks at the actual sequencing files. And they see if the sequencing quality is good, if it passes our thresholds, and so on.
And all this is manual, or this curation pipeline?
Part of the curation is automated. So I mean, especially, well, it depends in which archive the data are. So when they go to ArrayExpress or BioStudies, that is EBI run archives, there we actually know what kind of information gets recorded. So we have automated pipelines that check for these terms and helps the curation. GEO is not so consistent, their metadata are not as consistent. So there we still have an automated part, but a curator in many cases needs to go and look at it as well.
I have other questions, but maybe yes.
I have many questions as well, so I will not ask all of them at once.
Okay.
Thank you very much. This was very interesting. I will start with a very basic question. So the Expression Atlas is not an archive, right?
No.
So what makes it different from GEO and anything else?
Okay. So an archive is where the experiment is deposited as is. So someone who wants to publish their paper and they need to have their data in the public domain and give an accession number to show to the journal. Yeah. They go into an archive. So, and the data set stays there. So you have the raw data, the processed data files submitted by the authors and links to the paper and so on. So what we do, Expression Atlas is an added value database. So we take the raw data and the metadata from the archives and we reanalyze the data. And then what we show is the results, the experimental results and the metadata in a searchable way. So the value we add is by doing the analysis and bringing the analyzed data together and making them searchable.
Is it fair to say that the results of the analysis are also a data set?
Yes, the results are also a data
And
set.
do archives store analysis results as well?
So they do store in a different way. They just store the files. And these are the analysis done by each author. So they're not, the analysis might not be consistent across data sets in an archive. It depends on what tools the authors used, which bits they did manual and so on. And also the analysis results are not being indexed. So we index them together so that you can search across experiments, across data analysis results.
And do you do any check whether your results confirm the results of the original authors if they are published?
Yeah, so we do that periodically. We go back and check the correlation gene expression between our results and the original ones. And another thing we do is send an email to the submitters once their data set has been reprocessed, especially for those submitters submitting to EBI, to the EBI archives, so that they can also check for themselves and be aware that their data set has been reanalyzed. And generally, I would say that we find consistent results. You will find the top genes to be there. They might not be in the same order, but they will be significantly differentially expressed, for example.
Okay, I will stop here. This is one group of questions.
Okay.
There's a question on the chat. So it's from Fotis. Thank you for the presentation. As I see that the EBI expression atlas is interconnected to databases like EMBL and open targets, I would like to ask whether it also retrieves and curates drug protein association with these databases.
Okay, so no, we don't retrieve these associations. So what happens is our data feed into open targets, and there in the open targets platform, the gene expression data come together with the drug and protein associations that are also there from Campbell. So it's a relationship in an opposite direction.
This pipeline that had two branches that, well, I did not quite understand the right branch, but
This one. This one.
Yes, mostly this. Perhaps I missed it somehow.
So basically, there are two different ways of dissociating and evaluating cells on there are two different major protocols for doing single cell RNA-seq. So one is the plate-based studies where the cells are mechanically sorted and put on a plate in its different position. And the second one is the droplet-based, where the cells are attached into oil droplets, and they're barcoded, so you can have multiple cells, and this is higher throughput. But that means that the first bit of the analysis needs to be slightly different. And that's why we have a tool, Alevin, that performs the processes that you need for droplet-based studies, which is really, it deals with the barcodes. So these are not, the cells here are not barcoded. So here we need to resolve the barcodes and demultiplex, as well as, we have some droplets that are empty. We need to remove it. We might have doublets. So two cells sequenced in one droplet, for example. These also need to be removed because they're not informative, which means we have to have two different parts of the pipeline based on what technology has been used.
Thanks.
No problem.
Thank you. So moving down the pipeline, you talked about proteins. Does that mean that you are also considering isoforms?
So we are starting to consider isoforms on bulk expression atlas and on the gene expression side. And that's because yeah, we can do it there because the whole transcript is being sequenced. For single cell, we cannot do it yet because especially most of our data come from droplet-based studies, and these they don't sequence the whole RNA transcript, only the 5' end, the one end or the other end. So you cannot really see the splice junctions and so on and in further. But there are new developments on using nanopore sequencing that at the single cell level that will enable this kind of detection.
So single cell technology at the moment cannot distinguish the isoforms, is that?
No, not really. I mean, the smart protocols could distinguish, but they're not as high throughput, and there are not that many data sets around, as many as droplet, but the droplet ones cannot distinguish.
So more generally, if somebody in the bulk case, if somebody had some experiments and they were, I'm actually thinking of a particular project that we are involved in, and they are measuring different isoform expressions at different parts of the body. How would they contribute that? So we're talking about a European project, 5 different teams. Each team is measuring isoform expression in different tissues, and they want to contribute that to the expression atlas. How would they do it?
So I think they would need to contact us first, so that it's RNA-seq, bulk RNA-seq. So if we support the type of experiment, and it matches also the analysis we do for isoforms, we basically align them to alternative transcripts in ensemble. We don't do de novo quantification. So if they want de novo, then we cannot do it. But if they're
by de novo, you mean a new isoform?
Yeah, just assembling them and predicting what are the isoforms. What we do is we take the data and we align it to ensemble transcripts, and then we can distinguish between the different transcripts.
Okay, that's interesting to know. And individual researchers, before archiving the data in Geo or anything else, they can come to you and say, I have this data, and
for analysis?
Maybe they have analyzed that themselves, but to give it to you to add to the expression. So
what we do, if that happens, we have had some people who want to do that. They can still submit it through the EBI archive. It goes in as private, then it gets processed for Expression Atlas, and it's also private, so the particular experiment. And so what some people did, even they took the URL with a private key from Expression Atlas and put it on the letter to the journal where they submitted their work for the reviewers to have a look. So yeah, this is possible. And then it becomes public when their paper gets published.
So that also provides a way of archiving data.
Well, not really, because we don't give accession. So it does provide a way of archiving, because they will go to the archive first, the EBI archive, and this process will be streamlined into Expression Atlas.
Okay, great. I will take a pause here in case somebody else is.
Yes.
I noticed that you mentioned some ontologies that are used to annotate the data sets. And since this is a long running project, do you have cases where you need to re-annotate or update the annotations of previous studies, or due to this evolution of the
so yes, that's a good question. So yes, first we use a lot of different ontologies, like cell ontology, anatomy, EFO, environmental factor, and so on. And these are managed by another team at the EBI, the SPOT team, by my colleague Helen Parkinson. And they have developed tools that enable us to re-annotate the data. So we run before it's released, before we release, we run the ontology update, basically that looks at the terms we have, updates them with the new terms, if possible, from the kind of ontology facilities by the other team. And then a curator looks that they still make sense because there are errors as well. And then we continue with release. So we have that there. Also, so the ontologist team at EBI has also developed like a service where a curator getting a new experiment and they want to search for a term to see if it exists. If not, they suggest the term and also its place in the hierarchy. And then the ontologist team has it as a task to extend the ontology or communicate. Some ontologies are built by other consortia, so they communicate with them to have the new addition. So we contribute to that as well.
So based on this process, I guess this would result in some cases, especially for past studies, that we have some null values for some terms, for example, that previous studies may didn't track. I don't know, is this possible, I guess? You
mean we have new terms for new values for terms that before they were not tracked? Yes, not tracked. For example, a study 10 years ago may didn't track this specific information that on which we are now very interested, but then maybe we didn't have this information. So if we reannotate this study, maybe this information is not available in the paper. Is this any case, or is this the format always the same in the primary data, in the raw data, so we can always extract the information? I think we can always extract the information because once we get if I understand well your question, once we get a study, a curator will annotate everything with what exists on the ontologies at the time. So I don't think if it's something important that we cannot find an ontology identifier, we'll create one and suggest an addition to the ontology. If it's not important, then we don't track it and we don't go back. But it's probably not important for the analysis. So yeah.
I have also one more question.
Okay.
You mentioned at the end, one of the tasks that you want to do is text analysis on the bio studies, right? Can you say more things about that? Well, in which part you want to use it? It's the papers that you submit with a study that you want to analyze, or
yeah, so
else?
basically yeah, on bio studies we have the different well, we have metadata files and links to the papers, and what we would like to do is be able to extract the experimental information using text mining from the paper.
Now this is done by hand by the curators?
Yeah,
okay.
It's manual.
Okay, good.
Thank you. Moving to the single cell work that I don't really know much about. You talked about cell types, and you talked also about the periodic table, which I guess maps the cell types, right? How much progress has been made there? I mean, how mature is this work? How do we know the cell types? Do we get new cell types all the time?
So the major cell types are known. There are some rare ones that have been discovered and are being discovered using single cell sequencing. What is also really interesting is to find the different cell states of a cell type, because they're not static and they differ. They go through phases. For example, they differentiate into another kind of cell type and so on. And with single cell sequencing, we can find all these kind of in-between states as well.
So it's not completely stable yet. There's still a lot of work.
The main types of cell types are stable. We know what they are, yes, but it is in progress. There's no periodic table yet. It's just like an image. It's the goal. And a lot of them have been characterized. Some of them were already known. They have been better characterized. And some rare cell types have been also discovered. So it gets completed, and also states in between cell types are being discovered as well and analyzed.
How about cancer cells? I mean, do they belong to one type, to many types?
So that's a good question. So if you take the cells from a tumor, we call them, malignant cells. They might look particularly more like a particular cell type where they started from. For example, there's a lot of differentiation within the different malignant cells. So I think these are not studied particularly in this way. And that's why it makes it different. It makes it really difficult to relate single cell profiles of cancer patients, even though they had the same cancer type.
By cancer type, you mean the known cancers
like
of
breast
lung
cancer.
cancer? Yeah.
For example, if
Would-
you have two
would-
patients of breast cancer, even if it is the same type of breast cancer, when you want to integrate the single cell data, it's really difficult because the gene expression differs so much
What does that mean for the atlas,
on the
for the work that you do?
for the single cell expression.
Because there's a lot of work on single cell, I understand, on cancer, right? There are probably a lot of data around out there.
Yeah.
Tumor cells.
So what you can do, a lot of work on the, so we do have tumor profiles in the single cell expression atlas. We use the annotations, the cell type annotations that come with the primary authors that they submitted. We just do the reanalysis ourselves. But the labels of the cell types are as determined by the original authors. And anyone can search for these terms and find the profiles and the gene expression. Now, yeah, I'm not sure exactly what you mean, what it means for the atlas. That's how we have dealt with this.
Yeah, basically, my understanding of the atlas is that you need to have the types beforehand in order to organize it. First of all, to link it to some ontology. I mean,
Yeah,
if you don't have
we
the
do.
types, you cannot.
Yeah, so we request the cell types. So if I go to this slide with this one, so this is what we call inferred cell type as inferred by the submitters. So we request the labels basically of the cell types for each cell that we take in, because this is not a very automated process to determine which of the clusters, what kind of cell types they are. And it requires a lot of expert knowledge that we cannot reproduce in the team. I mean, there are some tools that train on already annotated data to automatically annotate new data types, but we have used a few and they're not as reliable yet, I think, because the reference data are not enough at the moment. It's still not a very widespread technology. It's very expensive to do single cell RNA-seq. But in the future, it will be able to annotate your single cell data set based on a previous one. But at the moment, yeah, we just take and we map them to ontologies.
And you map them to the ontology. So you take the cell types that have been declared by the authors, and you have to map them to some known cell types.
Yeah, so there's a cell ontology, cell type ontology, and that's what we use. So it's we look for the closest term, we see if it can cover, is it a synonym, and so on, and we add ID.
I think this is also an interesting NLP kind of ontology and
Yeah.
NLP kind of work, because I bet a lot of people are using, especially now that, you know, the whole situation is not quite stable, they will be using different names for the same thing.
Yes, they do. We find that a lot. We use different names for the same cell type and so on, because it's not stable. So yeah, I agree.
And I have heard of people who are working on single cell work. I know one particular group that I know of a bit more. They're trying to build, you know, an atlas of cancer cells or something.
Yeah.
Like the one that I'm
H-STAN.
Steinart's. I don't know if
Yes, I know. Yeah.
Does this atlas have anything to do with your atlas? Probably different.
So I think it's loosely connected to the Human Cell Atlas, and we are also part of the Human Cell Atlas. So because the Human Cell Atlas is supposed to produce the healthy atlas of the cells, like in a health state. And then there are these other disease atlases that are happening at the same time, and the Cancer Cell Atlas is one of them. So basically, from my point of view, from the EBI, from the expression atlas point of view and single cell expression atlas, these data, once published, they will come into the resource for the whole community to search through them. So that's the vision.
I have one question because it's related. Does in the Human Cell Atlas or in your work, does the frequency of the cell type play a role?
You mean the abundance?
Yeah.
It does play a role. So, for example, in tumors, you may have abundant immune cells within the tumor, and in other tumors you may not have. That means you can target with immunotherapies those ones that have abundant, immune cells within infiltrating the tumor, but you cannot on another tumor where you don't see as many, because it won't be effective. So yeah, it matters. It also matters on cell-to-cell communication. So there's like a significant research line that is really taking off at the moment. Some colleagues at EBI, for example, are working on that, on identifying what gets secreted in one cell type and it gets detected in the adjacent cell type and how they communicate. And there also abundance must play a role as well to, how much of the compound is being excreted to be sensed by the other cell types and so on.
Okay, thank you.
One more question from there. So it's from Aspa Vozi. Thank you very much for the very interesting talk. It is also possible to combine data from different species. And if so, have you tried to integrate it in your analysis?
Yes, it is possible. In fact, my PhD student, so this is on the research side. Here, I only showed what my service team is doing. But one of my students is actually investigating if we can integrate on the UMAP space data from different species, and how well that integration works according to their evolutionary distance. Where does it break down? And you need to use other methods and so on. And we have a preprint on that. So if you're interested, I can send it and you can have a look. But yeah, it's possible. It's not on the service yet. It's still a matter of research. But we hope that at some point it can become part of the service.
Do you have any other questions? No, I think that's enough.
Okay.
Already. Thank you. Let's thank the speaker again. Thank you very much.
Thank you.